Think of your body. You have cells that make up your protective skin, the body's largest organ, cells that form your heart tissue, keeping blood flowing at all times, and neurons that allow for electrical impulses to be transmitted in the brain. Each is built from different kinds of cells, but at one point, all of them came from the same type of early flexible cell, a stem cell. In the 2024 paper published by the Le Bon Lab, they wanted to look at this question of development within two evolutionarily connected species. About 500 million years ago, backbone-containing organisms known as vertebrates split into two groups, first being jawless, the cyclostomes, and second, the jawed species, the gnathostomes. Here they used African clawed frogs, a typical model organism, and lampreys. Both species have specialized neural crest cells that can form many parts of the body, but scientists wondered, did this flexibility evolve twice, or did it come from a shared ancestor? At the very start of life, blastula cells, which form from the zygote undergoing cell division, are pluripotent. They can differentiate into any cell type. However, neural crest cells, as seen on the right, seem to regain this ability. To look deeper into the embryos, researchers first used in situ hybridization, a method that stains cells where certain genes are switched on. Basically, they could see which genes were active in the lamprey's early embryos, and this was followed by RNA sequencing to measure all the gene messages in these cells, comparing them to frog embryos. They found that both lamprey and frog embryos use nearly the same set of genes, including TFAP2A, ZIC, and MYC in their early cells. This means the genetic network behind pluripotent cells and neural crest cells evolved long before the split between jawed and jawless vertebrates. One key gene, POU5, acts like a switch keeping cells flexible. In lampreys, it turns off before neural crest development, but in frogs, it stays on, helping create new tissues. Overall, this research reveals that the genetic tools making stem cells flexible are far older than we thought. The same network that once helped ancient vertebrates form new features such as jaws still drives how our cells develop and regenerate today. Evolution's innovations never vanished, they were just repurposed within us.